Hello students, and today we're going to continue Chapter 17, the urinary system. Welcome back to your survey of human anatomy and physiology. In this picture here you can see one of the three parts of the nephron board, the model that you need to be familiar with, and having a labeled key for that model will help you to study a little bit. And today we're going to look at how urine forms, urine formation, and the glomerular structure. And we remember this was the renal cortex and the renal medulla, and the nephron with the nephron loop and the collecting duct. Okay, it collects urine, which goes through the renal papilla down to the minor calyx, to the major calyx, to the renal pelvis, and to the ureter, to the urinary bladder, through the urethra, and outside of the body. Let's talk about urine formation. It really happens in three steps. They're a little bit complex, and we're going to go into those more in detail. The first step is glomerular filtration. The second step is tubular reabsorption, and the third step, tubular secretion. Now, we already went over the structures of the glomerulus, the renal capsule or the glomerular capsule here. Here's the end of the proximal convoluted tubule, so the glomerular capsule gives rise to the proximal convoluted tubule. And entering this glomerular capsule is the afferent arteriole, and then here's the glomerulus and the efferent arteriole. Now I want you to look closely at this picture and notice how the afferent arteriole is larger in diameter than the efferent. You could say afferent or efferent. Either way is correct. Okay, but the afferent arteriole is larger in diameter. Efferent is more narrow in diameter. Now, what is the effect here? Okay, so you're delivering a larger volume of blood to this network of capillaries of the glomerulus, and as it comes out, if you have a more narrow blood vessel, then this is going to increase blood pressure. Okay, so the first thing that happens in urine formation is glomerular filtration. Because you're delivering more blood here, less blood here, it raises the hydrostatic pressure and filtration happens. It's sort of like delivering the water to your house. It comes from the main and maybe a 2-inch pipe, and by the time it gets to your shower, it may be delivered on a 3/4 inch pipe. And as you go from a larger pipe to a smaller pipe, it increases water pressure, so you get a good water pressure in your shower. Well, this is sort of the same concept, where you have a larger vessel and a series of capillaries in that glomerulus, and then the efferent arteriole, a smaller in diameter vessel, so you're increasing hydrostatic pressure here. And when you increase the hydrostatic pressure inside the glomerulus, remember we had, this is a capillary that is a single or simple squamous epithelial layer of cells. So it's simple, meaning one layer of cell, very thin, and within that wall, that capillary wall of that glomerulus, there are pores or openings called fenestrae here at where these cells are joined together. and that fenestrae allows certain molecules or substances to pass through in that glomerular filtrate. As that blood passes through under pressure, it's going to allow for the pushing out of certain substances. Now, things that are large, like blood cells and large proteins, will not filter out. They're too big to go through that fenestrae, so they remain in the bloodstream. But smaller particles or molecules are allowed to leave into and dump into that glomerular capsule, and so that's the first step in what will eventually be urine. Initially, it is not called urine; it is called glomerular filtrate. The definition is a liquid part of the blood is force filtered through the walls of the glomerulus, and that's glomerular filtration. You have the podocytes and the fenestrae in that capillary of the glomerulus. The greatest force in glomerular filtration is hydrostatic pressure, glomerular hydrostatic pressure. Hydro, water, and hydrostatic pressure is the same thing as we talked about before, blood pressure. So you have a certain amount of blood pressure coming from circulation of the blood from the pumping of the heart, but we're a pretty good distance away from the heart. So as you get further away from the heart and you went from an artery to an arterial, the blood pressure, hydrostatic pressure from the heart is dropping, so you have the larger in diameter afferent arteriole, and then the glomerulus, and then the smaller and more narrow diameter, a smaller tube for the efferent arteriole, and that increases that hydrostatic pressure, and some from the pumping of action of the heart. Liquid enters the glomerular capsule, and this is called glomerular filtrate. What came out of the glomerulus is the glomerular filtrate. The glomerular filtrate has a composition very similar to tissue fluid or plasma. It contains useful materials and wastes. There's water, glucose, some amino acids, a very small proteins like albumin, but the larger proteins stay within the blood vessel. Urea, uric acid, sodium, bicarbonate, phosphates, and chlorides are examples of materials that are passed into the glomerular capsule by hydrostatic pressure, and it is found within that glomerular filtrate. Okay, let's look at this for just a minute. Here is the glomerular filtrate in this graph, and look, 180 liters in 24 hours, but yet urine output is anywhere from close to half a liter to 2.5 liters in 24 hours. So all of this filtrate is produced, and here's urine. So this isn't, glomerular filtrate is not urine yet. You have this fluid filtrate has to be processed, because if we were dumping all these things, we would be in pretty major trouble. Okay, glomerular filtrate rate, or filtration rate, or GFR. Normal filtration rate is about 125 milliliters per minute. Glomerular hydrostatic pressure is how it happens. It's the most determining factor. The changing of the size of the afferent, the diameter of the afferent or efferent arteriole affects the glomerular hydrostatic pressure. As pressure increases, filtration rate increases. It's directly proportional to each other. So glomerular filtration rate and hydrostatic pressure are pretty much equal in that as the hydrostatic pressure increases, filtration rate increases. It has sympathetic nervous stimulation or no sympathetic nervous stimulation. Okay, we had discussed earlier that all of this is controlled. Homeostasis in the body is controlled in two ways: nervous stimulation and hormones. The same is true for the urinary system, the action of the kidneys to form urine. In our blood vessels, sympathetic nervous system constricts the tunica media. Remember the layers of the blood vessels? That middle layer is made up of smooth muscles and it's under nervous system control, autonomic nervous system control, and the sympathetic response would be constriction of that blood vessel. And that's what happens with the afferent efferent arterioles. So sympathetic nervous system constricts the afferent arterioles. So what happens when it constricts? Constricts means that it becomes more narrow. So remember we just talked about the afferent being a larger diameter than the efferent, and because of that it increases the hydrostatic pressure between those two vessels, because you have a larger amount of blood being forced through from the afferent and a smaller amount on the other end, so it sort of backs the blood up, and so filtration happens. Well, if you have sympathetic response and it constricts the afferent arteriole, it is decreasing the diameter of that vessel, and if you're decreasing the diameter and the efferent is smaller in diameter, they're going to be more equal to each other, so you're going to have less hydrostatic pressure. Glomerular hydrostatic pressure less means that you will have less GFR, or glomerular filtration rate will be less. Okay, so the sympathetic stimulation would constrict the afferent arteriole. If you do not have sympathetic stimulation, then it remains in its normal dilated state. Okay, so that's one thing that happens. Now let me throw this in here real quick. The sympathetic nervous system can not only affect the afferent arteriole, but it can affect the efferent arteriole. So say the afferent arteriole stays the same size, larger in diameter than the efferent, but the efferent constricts. So if the afferent arterioles, its normal size, larger usually than the efferent, but now the efferent is constricting, it's going to become even more narrowed. So you're going to have a higher glomerular hydrostatic pressure. With a higher glomerular hydrostatic pressure, you're going to have a higher glomerular filtration rate. So that's two ways that the sympathetic nervous system can affect the hydrostatic pressure in those vessels. You have autoregulation. The vasoconstriction increases sympathetic impulses, which decreases glomerular filtration rate. And we just talked about that. So vasoconstriction, the constriction of the vessel is what that word means, vasoconstriction. If you're narrowing that passageway, reducing the diameter of that vessel, it's going to, that afferent arteriole, it's going to decrease the filtration rate. Okay, so remember, because it was larger, it was dumping a larger amount of blood, or sending a larger amount of blood than can pass through that smaller diameter efferent, you constrict it, and it's going to decrease the filtration rate by causing afferent arterioles to constrict. A second autoregulation system is the renin-angiotensin system, and that's what's shown here. The liver will produce a hormone called angiotensinogen, okay? And in response to the production of angiotensinogen, the kidney will secrete the hormone. Remember that, let's go back for just a second. Remember that equilibrium, homeostasis in the body occurs from action of the nervous system and hormones. So the kidney produces the hormone renin, and it reacts with angiotensinogen. And when renin and angiotensinogen react to each other, it forms a compound called angiotensin 1. And then an angiotensin converting enzyme is, as this blood passes through the capillaries found in the lungs will release angiotensin converting enzyme, and that changes that angiotensin 1 to angiotensin 2. And then when that gets back to the kidneys, it's going to cause all these things. Okay, vasoconstriction, which is going to decrease the hydrostatic pressure, decrease the GFR, or the glomerular filtration rate. In response to this chemical in the blood, there is going to be the release of aldosterone. Do you remember where aldosterone comes from? Aldosterone, remember that sitting on top of that kidney is the adrenal gland, and from the adrenal cortex aldosterone is released. So increased aldosterone secretion and increased ADH secretion and increased thirst. It is released into the bloodstream, and then you have the stimulation for these things. And then a third way of regulation of the filtration rate is atrial natriuretic peptide, and this is produced from or released by the heart. Okay, we'll look at each one of these more closely. Vasoconstriction we already talked about, narrowing of the blood vessels, the afferent arteriole to decrease filtration rate, increased aldosterone. Do you remember the action of aldosterone? If you don't remember, you need to go back and review that. This is the hormone that helps you maintain your sodium levels, and it gets rid of excess potassium. So it holds on to or conserves sodium, and it secretes potassium. Okay, causes the secretion of potassium. Increased ADH, antidiuretic hormone. Where is antidiuretic hormone secreted from? If you said posterior pituitary, you are correct. Antidiuretic hormone. And then there is an increased thirst. Tubular reabsorption. First we had glomerular filtration, and then we have tubular reabsorption. All those things passes out of the bloodstream and into that filtrate, dumps into the glomerular capsule. That's glucose, water, sodiums, some amino acids, uric acid, urea. A lot of things are dumped into this filtration. in this glomerular filtrate. The process by reabsorption is the process by which substances are transported from the glomerular filtrate into the blood of the peritubular capillaries. Now, here is the tubule. We already talked about the glomerular filtrate. It dumps into the capsule here, which dumps in the proximal convoluted tubule. Now, we need to process this filtrate because we can't lose all those substances. We need to pick these substances back up. So what happens, they leave the proximal convoluted tubule and return to the peritubular capillary or the bloodstream. So a lot of the important things we need to maintain or hold on to is reabsorbed into this peritubular capillary. It is carried out of or into the peritubular capillary by carrier molecules in a process called active transport. Now, we talked about diffusion. Just a little review here. Diffusion, as we said before, is the movement of molecules from an area of high concentration to an area of lower concentration. Now, it is a natural physical process that does not require any expenditure of energy from higher concentration to lower concentration. In this process, it's just the opposite. Things are going to be moving. You already have a high concentration of these things in the bloodstream, and now you're moving more of that same substance into the bloodstream. So you are moving against the concentration gradient. You are moving from a low concentration of these substances to a higher concentration of the substances. So it would not be diffusion. It only happens in a process called active transport. Now, active transport requires the expenditure of ATPs. Remember, we talked about ATPs before, so it uses energy. On the surface of this proximal convoluted tubule, this renal tubule, there are microvilli. Looks like a brush, and this microvilli increases the surface area as we have discussed before. And the so with that microvilli or that brush area on the surface of the proximal convoluted tubule, it allows for more surface area, so the things that have been filtered out of the blood will be carried out of the tube and is allowed to move into the peritubular capillary. An example would be glucose. You lose quite a bit of glucose in this filtrate. Okay, well, you need glucose for metabolic activities all through the body, so you need to maintain that glucose level in the bloodstream. So you need to restore those levels. So through active transport, that glucose is allowed to leave the tubule, the proximal convoluted tubule, and you may have a little bit occurring in the all through the tubule, but most of this happens in the proximal convoluted tubule. But a carrier molecule is required to take that glucose back to the bloodstream. So it's transported out of the tubular fluid through the epithelium into the interstitial fluid and then back into the bloodstream. And it is moved by a process called active transport. Active transport requires energy. Glucose is present in the glomerular filtrate, but for the most part is absent in the urine. You may have a little higher or a little bit of a glucose level in urine if you were taking a test and testing for glucose if you just ate a meal with a high carbohydrate content, because you can only you have only so many carrier molecules that can carry the glucose, so you can only move a certain amount of glucose back into the bloodstream. That's actually called renal plasma threshold. And if you can't carry it all back in, then you may have a little bit of extra glucose in the urine. Okay, just a little FYI there. But substances that are needed to maintain homeostasis in the body are reabsorbed. They leave the tubule and go back into the bloodstream from the glomerular filtrate, reabsorbed into the bloodstream in the peritubular capillary. 99% of the glomerular filtrate will be reabsorbed. It occurs mainly in the proximal convoluted tubule, but it can happen in other areas of the renal tubule, but most of it happens in the proximal convoluted tubule. Has the brush border with that microvilli, and it's moved by active transport opposite of diffusion. It is moving from an area of low concentration to an area of high concentration. Materials reabsorbed include glucose, as we just talked about, water. You need to maintain, if you do not reabsorb that water, then you're going to drop blood pressure, you're going to not have the blood volume that you need, and so it's very important to maintain your the correct amount of water in the bloodstream, or you become dehydrated. And so also urea, proteins, creatine, amino acids, lactic acids, citric acids, phosphate, sulfate, calcium, potassium, and sodium. We're going to mention like lactic acid, citric acids. These are high in hydrogens. The definition of an acid is a high concentration of hydrogens as opposed to hydroxyl ions, and we'll look at that again in just a minute, but note that it would be fairly acidic here. The water follows sodium. I mentioned before several times that if you have solute in a solution, it has taken the space where you could have water. So wherever the solute is greater, then the water will follow it. Wherever sodium goes, usually water will follow it. So water follows the sodium by osmosis. Review: the definition of osmosis is the movement of molecules from an area of higher concentration to an area of lower concentration. Water molecules through a semi-permeable membrane. Osmosis. Okay, so water follows the sodium by osmosis. The regulation of that urine concentration. Here we have, ADH causes water to be reabsorbed and forms a concentrated urine. So the urine is either going to be diluted or concentrated. Alright, so we remember that we have glomerular filtration here. And then we have the filtrate, the convoluted tubules, where you have the reabsorption happening, and so you lose the water through that proximal convoluted tubule. This is the descending loop and the ascending loop. Now, the ascending loop and the distal convoluted tubule do not allow water to pass through it. It is impermeable to water, meaning that permeable means it allows it to pass through, impermeable means that it will not allow it to pass through. So here in the proximal convoluted tubule and in the descending loop, limb of the nephron loop, you can water can pass out, okay? And it does. If you're losing sodiums here, water's usually going to move out with the sodium, and it can be reabsorbed by the peritubular capillaries. Okay, so if this happens here, you will have a dilute urine because some water's lost here, but only a certain amount. If if no more is lost in the ascending loop and in the distal convoluted tubules, then you're going to have dilute urine. Okay, so this is the collecting tube and or duct, and the result would be a dilute urine. Now, what makes, look at this picture. Well, I just said that it was impermeable to water, but over here it's showing the water is leaving. It can come out here, but it doesn't usually come out here. Antidiuretic hormone causes water to be reabsorbed and forms a more concentrated urine. Okay, so a little water is reabsorbed in the peritubular capillaries here, but no more here because this is impermeable to water. But the action of the antidiuretic hormones makes this ascending loop and the distal convoluted tubule permeable to water. So that is the action. Here's the target cells for that hormone, antidiuretic hormone. Okay, so if you are if water is allowed to leave the tubules and be reabsorbed by the it would be in the interstitial fluid, and then it would cross over into, by osmosis, go back into the peritubular capillaries picked up by the bloodstream. So since that water is being picked up by the bloodstream again, it is no longer over here the filtrate, and as it gets closer to the collecting duct, it becomes urine. So this urine is losing that water, being the water will be found in the interstitial fluids and picked up by the peritubular capillaries and return to the bloodstream. So the urine is going to be more concentrated. So what happens, antidiuretic, you know, a diuretic causes you to urinate more. Antidiuretic less, because you are you're maintaining that water level in the bloodstream, in the interstitial fluids, in the plasma, and so it's taking it back from this filtrate, and so the urine will be more concentrated. Okay, now remember that you do have some, you are losing sodium here, and so this is happening through osmosis, and it's called colloidal osmotic pressure, because sodium, all these substances that pass through the glucose, water, urea, proteins, amino acids, calcium, potassium, sodium ions, all those things in that interstitial fluid would be solutes. sodium, wherever the sodium goes, you know that the water's going to follow. So you have the, let's talk about countercurrent, the countercurrent mechanism. Now, without the ADH, remember that here we have the glomerular filtrate. It dumps into the glomerular capsule that dumps into the proximal convoluted tubule. The proximal convoluted tubule and the descending limb of the nephron loop is permeable to water, so some water is reabsorbed here or passes through that tubule, goes into the interstitial fluid, and then some is reabsorbed by the tubular capillaries to the blood plasma and bloodstream. Okay, so the ascending loop, again, is impermeable to water. And what did I say that meant? This is permeable. It allows it to pass through. Impermeable means it does not allow it to pass through. Just another tidbit, the proximal convoluted tubule and the descending loop are a little less permeable to these electrolytes, but you will have some passing, but a little less over here. So anyway, it is permeable to water over here, but in the ascending loop and in the distal convoluted tubule, it's impermeable to water, so you're not losing any water here. Here we have sodium being released. It's allowed to pass through. Now, look at this little plus sign. Sodium is a positively charged ion. By the way, an ion is a charged atom. Okay, so we have an atom of sodium here. It has a positive charge, so it is a positively charged ion, or an ion is a charged particle. This just happens to have a positive charge, and this is moving out because it's permeable to this sodium. And since this is positively charged, it's going to bring along with it a negatively charged ion. You know, negative and positives attract, so it's going to bring along with it that chloride. What's sodium and chloride? Sodium chloride, salt. Okay? Wherever salt goes, wherever the sodium goes, wherever salt goes, usually water will follow. But wait a minute. This is impermeable to water, so no water is lost here. But since this interstitial fluid is now becoming more salty, it's going to attract the water from over here. The proximal convoluted tubule and the descending limb of the nephron will lose the water because it's being attracted by this salt, by this solute. Okay, just the easy way to remember that happens is salt sucks. Okay, it actually draws that water. Why? Because of osmotic pressure, because wherever you have these ions, you could have water taking that space, but it's taking up space, so the water's in lower concentration over here and higher concentration over here, so it will move from an area of higher concentration to an area of lower concentration. You're not losing any water here because this is impermeable to water, but you are losing the water here. So what's happening is this countercurrent mechanism. Think of it moving counterclockwise, and that's why it's called the countercurrent mechanism. So here we're losing some sodium chloride. This is becoming a little salty, then more salty, then more salty. Now what happens so that you don't have all this salt in this interstitial fluid is that sodium chloride can be picked up by the proximal convoluted tubule, and or if it's needed in the bloodstream, it can be picked up through the peritubular capillaries, but it is the sodium content is maintained here by the movement of those ions, those molecules. But what we're looking at here is that counterclockwise current caused by the sodium and the potassium being given off here, the salt being given off here, and then the water here. The effect of ADH on the countercurrent mechanism: without ADH, the distal convoluted tubules and collecting duct are impermeable. But remember what happens with ADH, antidiuretic hormone: the distal convoluted tubule and the collecting duct become permeable. It allows water to move out. With antidiuretic hormone, the water is reabsorbed by osmosis into the hypertonic medullary interstitial fluid. Okay, into the medulla of the kidney. Okay, hypertonic means that it has more solute, so it's going to attract this water. So the water is going to move into the interstitial fluid with antidiuretic hormone, and when it's in the interstitial fluid, it can move in, be taken back up by the peritubular capillaries, and that's what happens. Now the vasa recta maintains the salty nature of the medulla area of the kidney. The fluid in the ascending limb becomes more hypotonic, meaning that it has less. Hypotonic has less solid. It is losing the sodium and the potassium, and so it's becoming hypotonic as compared to the interstitial fluid, because it's losing the sodium and potassium as the solute is reabsorbed. So remember that I said that it would be awfully salty here, and we can't have that salty situation because it would draw the water out, and we don't need it to stay real salty, so some of that sodium chloride is reabsorbed into the capillaries, some by the tubules, but into the capillaries. Okay, the fluid in the ascending limb becomes hypotonic as a solute is reabsorbed. The fluid in the descending limb becomes hypertonic as it loses water by the process of osmosis. And remember, these are the peritubular capillaries. Well, where the peritubular capillaries, you have these capillaries that are on the arterial sides, and then you have the venule sides. Alright, around that nephron loop, these sort of scoops of these capillaries that are around these peritubular, around these tubes, is called the vasa recta. The vasa recta helps to maintain the salty nature of the medulla. Okay, so we said that in the ascending loop and the distal convoluted tubule is going to be losing or giving up its sodium chloride, so this area is staying salty. Now it can't be too salty, so the vasa recta will allow for the movement of these ions back into the bloodstream, because you need sodium, a certain amount of sodium and chloride in the blood, sodium for different metabolic processes and actions, so you need a certain amount. So you're going to have some in the interstitial fluid, some's going to be reabsorbed, so The fluid in the ascending limb, since it is losing the sodium chloride, it is losing ions, it is losing that solute, it is called hypotonic. Less hypos, less tonic. It's going to have less solute as the solute is reabsorbed. Okay, the fluid in the descending limb, this is losing it, and then it's not being lost here. What's being lost here? Water. Okay, water comes out here. As water comes out here, then you're going to have more solute in this tubule. More solute makes it hypertonic. Okay, so the fluid in the descending limb is going to be hypertonic because it's losing water, so it's going to have more solute. Over here, you're maintaining the water, but you're losing, this is the solute, and so if you're less solute and more water, it's going to be hypotonic. And hypertonic, hypotonic situations will draw or give up the water. Okay, so hypotonic is going to lose the water because of the solutes out in this area. It's going to draw the water out. Okay, but again, the water can't come out here. So you have water, but you're losing the sodiums and chlorides, so it's going to be more hypertonic here. And so it will lose water by osmosis here and lose the solutes here. And the third step in that the beginning of that urine formation is tubular secretion. It transports substances from the blood into the peritubular capillary into the renal tubule. Now, we had glomerular filtration here. We had reabsorption, leaving the tubule, going to the bloodstream. Now we have just the opposite happening. It's leaving the bloodstream and going back into the tubule. Now, what's the purpose of all this? Well, it is maintaining homeostasis, the right amount of glucose, the right amount of water, the right amount of the substances. Now, we have to get rid of waste, so we've got to lose that uric acid. We've got to lose all the things that are toxic to us. It's got to be dumped back into the renal tubule so it can leave eventually in urine. So you have the secretion, moving substances from the peritubular capillary back to the renal tubule. So that is tubular secretion. So tubular, that's where it's going. It's being given up or reabsorbed by the blood vessel. Okay, so the blood vessel is secreting these substances and dumping it into the tubule. Transport substances from the blood into the peritubular capillary. The blood in the peritubular capillary is dumping it into the renal tubule. Tubular secretion, why is it important? It disposes substances too big to enter the glomerular filtrate. That would be like penicillin, other drugs that are too big that would not have been filtered out. It eliminates undesirable substances that have been reabsorbed, the urea and uric acid. It helps the body to rid itself of excess potassium. You need a certain amount of potassium in the bloodstream, and you've got to get rid of the rest. It helps to control the blood pH. And just a minute ago I said that a higher If you have uric acid, lactic acid, citric acid, it's going to be acidic. But you're also going to have free hydrogens in there. That's also an ion, a charged particle. So you've got to get rid of those hydrogens, or it's going to change the pH of the blood, and it's going to be too acidic. So you've got to get rid of the hydrogens. So those will be passed from the blood to the tubulars so it can go into the urine and be disposed of. Bicarbonates. Remember, that was one way of getting rid of those carbon dioxides. Bicarbonates are also released by the blood into that tubule fluid and in the urine and gotten rid of. And that potassium. You don't need to lose too much of the hydrogens or the blood will become too alkaline. To retain too much of the hydrogens and it becomes too acidic. So bicarbonate is excreted. If the blood is too acidic, it's going to give up those hydrogens and those the uric acid and citric acid and things high in hydrogens, and then the free hydrogens. It's going to hold on to, if it's acidic, bicarbonate. You might have heard of bicarbonate before as an antacid. A bicarbonate is an antacid, so when you go get that Tums, it is a bicarbonate and it neutralizes acids. So if you have a higher level of hydrogens, it's going to be acidic, so it's going to hold on to the bicarbonates and the potassium and get rid of the hydrogens. If it's too basic or alkaline, it's going to get rid of the bicarbonates. Okay. Now, this is a pretty interesting chart. This chart shows what is in our plasma, the blood plasma. Sodium 142 micrograms per liter. Okay, milliequivalents per liter. And potassium 5, calcium 4, so forth and so on, chloride, bicarbonates, sulfates. This is what is found in plasma. Look how it compares to the glomerular filtrate. So basically what is being given up through the glomerular filtrate, through the fenestrae, given up into that filtrate, is equivalent to what you would find in blood plasma. But we need to maintain these substances in our blood plasma, so we can't give up all this. Some things we need to get rid of more, and some we need to reabsorb, and that's what happens. So urine, you are getting rid, or you're reabsorbing some sodium. You're dumping more potassium into the tubule so it can be gotten rid of, and so urine will have more potassium than that plasma or glomerular filtrate. So in that tubular secretion, you would get rid of more potassium, get rid of more magnesium, get rid of more chloride, and bicarbonates may be reabsorbed to maintain the pH. Sulfates. Phosphates, you need to get rid of those. They're waste products, metabolic waste products, so that would be dumped into the tubule through tubular secretion so that we can get rid of that in our waste. But compare these numbers, it's really interesting. Glucose in the plasma, 100 in the glomerular filtrate, but usually you would not have glucose in the urine. If, like I said before, you have a plasma threshold. And what happens is that those carrier molecules in that active transport are carrying that glucose back to the bloodstream. It can only carry so much in tubular reabsorption, and that is called limited transport capacity. Now, limited transport capacity, it can only carry so much, and then we have the renal plasma threshold. And for a diabetic, what happens is that they are not producing enough insulin. And what insulin does, it helps your body to process the glucose. And we store glucose in muscles and then the liver in glycogen. Well, if you are not producing insulin, then we can't process, or the body cannot process that glucose. It's not stored, and so it's in the bloodstream. And so if you have limited transport capacity and renal plasma threshold, it can only hold so much glucose or carry it back to the bloodstream. So in the urine you would have a higher glucose content for a diabetic, because only so much can be carried back or reabsorbed by the blood. Okay, and tubular reabsorption. So a urine test would show a higher level of glucose in a diabetic. Urea, uric acid, and creatine. Look at the differences here. Pretty dramatic. So during tubular secretion, these waste products are given off. Urine composition. Normal components, about 95% water. Urea, amino acid. Urea is from a waste product from amino acid metabolism. Where does amino acid come from? Remember, we said proteins are made up of amino acids. So the metabolism of proteins, amino acid metabolism, urea. Uric acid from nucleic acid metabolism, whereas nucleic acid is found in DNA. Where's DNA found? The nucleus of all living cells has DNA, so nucleic acid metabolism, uric acid. Creatinine from creatine metabolism. When you are in oxygen debt and trying to make more ATPs, you metabolize ATPs or produce ATPs with creatine phosphate, but anyway, creatine metabolism. Traces of amino acids and glucose are found there, and electrolytes. Electrolytes would be your sodium, potassium, chloride, and those charged particles usually that we're just talking about. Volume is about a half a liter, close to half a liter to 2.5 liters per day. Normal output is about 50 to 60 milliliters per hour. Less than 30 milliliters per hour is considered kidney failure. So if you are not urinating, for a long period of time, then that's an indication that you have a problem with your kidneys. Elimination of urine. You have the collecting ducts. Remember that it dumps from the distal convoluted tubule to the collection duct that goes through the renal papilla to the minor calyx to the major calyx to the renal pelvis to the ureter, and to the urinary bladder, to the urethra, and then the outside world. The ureters, they're the tube between the kidney and the urinary bladder. They usually run in the same direction as the vertebral column. From the kidney to the urinary bladder, you have two of them. There's a mucus coat, there's the lumen, again a mucus coat, a muscular coat, and a fibrous coat, and surrounded by adipose or fat tissue. And remember, the renal pelvis dumps into the ureter. And the ureter to the urinary bladder. the location of the urinary bladder. It is found under the cervix and the uterus in the female, is posterior to the symphysis pubis, or the pelvic bone, front pelvic bone here, and the urethra is very short in a female. Okay. It is for temporary storage of urine. It's found just posterior to the symphysis pubis in the male also, and right above the prostate gland. The tube from the urinary bladder to the exterior body is going to be the urethra. Okay, so here's the ureter. The ureter comes into the ureter comes into the ureter, comes into the bladder in the lower portion of the bladder, the inferior portion on either side, and here's the urethra. This area here where the ureters come in and sort of meet up with the opening of the urethra is called the trigone. It's an actual structure where you have the opening, the orifice of the ureters, and that meets up with the orifice of the urethra. So the female to the external orifice is a very short distance. Okay, for the male, here is the ureter, the trigone, the prostate, and this is the prostatic urethra, the membranous urethra in this area, and then when you get into the penis, it's called penile urethra. Okay, so it's divided up into the prostatic urethra, membranous urethra, penile urethra, and then the external urethral orifice. Micturition is the elimination of urine. The bladder distends and stretches, and stretch receptors are stimulated, and the micturition center is activated in the sacral portion of the spinal cord. So initial nervous information is sent to the spinal cord. The parasympathetic nerve impulses causes the distressor muscle to contract. The distressor muscle to contract. And then that information is sent, nervous information is sent to the brain, and the need to urinate is sensed by the cerebral cortex. Voluntary contraction of the external urethral sphincter prevents urination. So a person holding their urine is under voluntary contraction of the external urethral sphincter. A sphincter is a round, smooth muscle. Now usually that round, smooth muscle is under unconscious control. You can't control the opening and closing of that sphincter muscle, but in this case you have voluntary control of the sphincter muscle. Now if a person is unconscious or has brain injury, or when a person is deceased, they lose control, and there's that muscle relaxes and urination happens. When the decision is made to urinate, the external urethral sphincter relaxes, the detrusor muscle contracts, which squeezes that urine from the bladder. The urine is expelled. By the way, the bladder has a layer of tissue called transitional epithelial tissue, and it's got different shapes of cells, and it gives that bladder stretchability, stretch and recoil. And that detrusor muscle, the detrusor muscle contracts and urine is expelled. Micturition. Okay, I wanted to go back to this autoregulation for just a minute and talk about ACE inhibitors. When a person has elevated blood pressure, hypertension, it is sometimes associated with excessive release of renin, followed by increased formation of vasoconstrictor angiotensin II, which causes the high blood pressure. So if you've heard of a person receiving an ACE inhibitor drug, what is happening is it inhibits the release of the angiotensin converting enzyme, so this isn't formed, the angiotensin II is not released, and so it helps to regulate that blood pressure by blocking the release of angiotensin II. So angiotensin converting enzyme, ACE inhibitor. Okay. That's the end of part 2.